Welcome to Brain Ponderings podcast. I'm your host, Mark Matson. Today I have Professor Alvaro Pascual-Leone he's a professor of neurology at Harvard Medical School. He's also director of the Center for Non-Invasive Brain Stimulation there, which is what we're going to learn from him today about. He's had a long-standing interest in non-invasive brain stimulation for more than 3 decades, I guess. And I guess we'll start first. Alvaro, welcome.
Thank you so much, Mark. Good to be here. Thank you for having me.
My understanding, you're from Valencia, Spain?
That is correct. Yeah, I was born in Spain, in Valencia. That's right.
And you got your MD, PhD in Germany, is that right?
Yeah, my parents decided to send my sister and I to a German school in Spain because they thought the education would be a good one. And I ended up getting a scholarship from Germany to study medical school there. And I did an MD, PhD program at the University of Freiburg with a PhD in neurophysiology, in very basic sort of neurophysiology.
Well, there is this stereotype. I think that's true, actually, that Germans have a history of being really good technically and developing precision instruments and both science and engineering. And of course, there was a Nobel Prize given for patch clamp technique, which is developed there. But then I'm from Minnesota. I don't know if you know that.
I did not know that.
Yeah,
so my wife.
I'm from Rochester, Minnesota. I went to Mayo High School.
Oh, so did my wife. You're kidding me.
What?
Yeah, that's so funny. It's a small world, Mark.
What year did she graduate?
Oh boy, I can't disclose that. I think.
What? Oh, you don't want to give me what's her maiden name?
Espinosa, Espinosa.
Okay.
My father-in-law was a neurologist at Mayo.
And, oh my gosh! Okay, I don't want to get
wife, too. No, yeah, it's okay.
Yeah, but still, this is interesting how the world goes, right? And these connections. One of my classmates was named Thor Sunt, Soralf Sunt III.
Sure.
And his father was a neurosurgeon, and now Thor's the head of I think head of thoracic surgery at Mass General up there in Boston.
Yes, that's correct.
So he was. I went to all the advanced science classes with him in high school.
That's a very small world. That's interesting. Yeah. I, of course, know and admire Dr. Sunt from the father and for neurosurgery and yeah, you're absolutely right. I ended up going to Mayo, then to the University of Minnesota. I did my neurology training there. I did clinical neurophysiology there. I met my wife there, and then we ended up moving to NIH, actually, to Washington, D.C., where I carried on the work that I had, in fact, started in Minnesota, the development of transcranial magnetic stimulation.
And okay, so you were first exposed to that in Minnesota, that technology?
I was.
Then at the NIH, you were, you have so many papers, I kind of scrolled through them, but going back there, you were incredibly productive. You were in Mark Hallett's very famous neuroscientist, neurologist, and you publish like crazy, looking at TMS and whatever stroke patient. So first of all, describe what TMS is for our listeners who have not heard of this. How does this technology work?
Yeah, so basically, it might be worthwhile to take even a step back and think of it from the point of view of brain organization. So what we've come to learn, in fact, ever since Cajal, the famous Spanish neuroscientist, and then evolving since then, is that the brain has these very specialized neurons, cells that make up the brain that Cajal was the first one in describing, but that they are engaged in networks, in circuits, and that those networks ultimately are the functional unit of the brain. There is a circuit in the brain, literally for everything we are able to do, think, feel, experience. Circuits in the brain account for our abilities, and alterations in the structure or the function of circuits in the brain account for the symptoms of disease. Yeah. And so now the question becomes, how can we query those circuits? How can we understand their function? And building on that, can we modify the activity in those circuits in order to have a therapeutic impact? And the traditional answer to both of those questions has been that there might be ways to do so invasively, using surgery, using approaches that put electrodes into the brain. And of course, that is incredibly powerful, but it's also risky. And so I became interested in the question, can one do the same? Can one query those circuits? Can we alter their function? Can we modify it for therapeutic intent non-invasively, without surgery? That requires knowing exactly how to target a given circuit, and then it requires bridging the skull and the scalp and getting to the desired location in the brain, which is not a trivial matter. But transcranial magnetic stimulation is one way to do that. And the principle behind transcranial magnetic stimulation goes back to a very famous scientist of England, Michael Faraday, who described the principles of electromagnetic induction in the mid-1800s. 1900s, 1830-some. And what Michael Faraday described is that if you pass a current through a coil, it generates a magnetic field, and that magnetic field can penetrate the scalp, and the skull reach the brain and induce a current in the brain. And that's exactly what we do with transcranial magnetic stimulation. So think about it as electrode-less electric stimulation of the brain via electromagnetic induction, but that's a mouthful. And so it became transcranial magnetic stimulation, although it's not the magnetic field that does the effect, but rather the electric field generated by the changing magnetic field that does it. And so what it allows us to do, sorry, just one last thought, what it allows us to do is to explore the relationship between activity in a given circuit and a given behavior, emotion, ability on the one hand. So explore relations between brain activity and behavior. And on the other hand, we discovered that it allows us to modify the pattern of activity in those circuits in such a way that may translate into a symptomatic treatment for disease. It doesn't cure the disease, but it reduces the disability.
Yeah. And some of the first studies that tried to, when the technology was being developed, my understanding is that the motor cortex was targeted because the idea was if you can stimulate those upper motor neurons that send signals to your muscles, then you can just see, you can see in real time a response.
Yeah, so it's a very interesting history. So there is a lot of efforts even well before that, including Bickford at the Mayo Clinic using attempts to use transcranial magnetic stimulation for various things. But there was a fundamental problem with the early attempts, which is that the critical thing to induce current in the nervous system that is sufficient to generate an effect, the critical factor is the rate of change of the magnetic field, how quickly the magnetic field rises and falls. And it wasn't until Tony Barker, an engineer at the University of Sheffield, that the discovery was made that that using a electronic switch called a thyristor, it was possible to pass the current from a bank of capacitors into a coil so fast that the magnetic field would rise and fall quickly enough. And with that in hand, Tony Barker tried to look for applications that offered new insights. And indeed, one of the first ones was in the motor system, because just like you described, by activating the motor cortex in the brain, it was possible to then assess the integrity of the connections between the brain and the muscles, and the time it took to go from the brain to the muscle, something that we call central motor conduction time. And so it was possible to explore how intact that connection was, how quickly it conducted, and in doing so, explore the cause for movement problems in patients with a variety of different diseases. And a lot of that work started in Europe, in the UK, in London, in Italy, and that's the interest that Mark Hallett, my mentor, had in using transcranial magnetic stimulation to explore the motor system, of which he was an expert. And I joined him with an interest of learning from him, but also with an interest of expanding the techniques to more behavioral cognitive applications. I was interested in things like language processing and speech and emotions. And one of the uncanny things we came to realize is that while a single pulse of TMS was enough to evoke a movement and therefore explore the movement, in order to disrupt things like speech, you needed repetitive pulses. You needed more than one stimulus at a time. And so that was the development, the technology development that I was involved in, and that John Cadwell ultimately enabled in the United States and led to what we now call repetitive TMS.
And also those early studies looking at the motor system, you're able to determine that, like, the spatial, how small an area can you stimulate, right? Because you got this.
That is correct. Yeah, we can basically move the coil, this stimulation coil around on the subject's head and evoke responses. And in doing so, we can do a sort of map of the cortical control of muscles in a very similar way as can be done in surgery, stimulating the brain directly. But this is non-invasive. Now, the important thing to emphasize there is that the ability to be precise with the stimulation has also significantly evolved in the last 20, 30 years. So we're now a lot more precise. We have tools and devices that are a lot more small in the area that they activate. And with that, the precision of that mapping has improved.
And so right, so you mentioned the brain has these neural circuits that mediate all our behaviors, emotions, and everything. And the core circuitry is excitatory neurons that use glutamate as a neurotransmitter, and those can be very long and extend between brain regions or all the way from the brain to the spinal cord and so on. And then inhibitory GABAergic neurons that kind of locally control excitability. Now, is it known, are both of those cell types affected similarly? And how do you can't really tease that out in a human. How do you determine?
That's a great question, Mark. Yeah, so we know that that in the brain there is a large number of brain cells that are exquisitely specialized, both in terms of the chemicals they use and in terms of how they connect with each other. Compared to that, a very precise and exquisite organization, TMS is a rather crude tool. It applies a pulse. It affects probably all the brain cells in similar ways, in a very somewhat non-specific way. And it's not possible, as far as we know, to be selective for one type of cells or another. It is, however, the case that depending on the state that a given brain cell is, the effect of the stimulation will be of one type or another. So the stimulation is imprecise and somewhat crude, but it's an interaction that ends up happening with the state of activity of the brain and the brain cells. And in leveraging that state dependency, we call it, we can make the effects more precise. It requires combining stimulation with certain behaviors or with other interventions. In order to understand better exactly what mechanism of action TMS has, one needs to take insights from humans into animal models and do work on animal models. And again, the cool thing about this technique is that it represents what I like to call a true translatable biomarker or intervention. So meaning you can do exactly the same intervention in an animal model, in a mouse, a rat, a cat, a dog, a baby, a young person, an old patient, a healthy or an elderly ill patient. So it's applicable in the same way across a large number of models. And that allows us to really start to tease apart the mechanisms of action. But there is no doubt that a lot more work is needed to fully understand that.
You've become interested in recent years a lot in brain aging. And of course, aging is a major risk factor for stroke and for Alzheimer's and Parkinson's. And in some of your early work and others, why don't you talk a little bit about the early work in stroke patients and seeing whether TMS with various stimulation parameters can have an effect in recovery, because when a person has a stroke, oftentimes they'll recover some function, not usually actually, recover some function, and they go through physical therapy and so on. That helps a lot. But the idea is that, yes, there are some neurons that are probably died and they're gone, but there are a lot of other neurons and neural circuits that just aren't functioning properly. And also there may be reorganization because it turns out there's this phenomenon of activity-dependent neuroplasticity that I started out as a developmental neurobiologist, and this idea that neurons that fire together wire together, and activity causes production of neurotrophic factors, and they promote their survival and synapse formation and so on. So why don't you talk about that idea in relation to stroke, and then maybe also depression and age-related cognitive impairment?
Yeah, absolutely. Thank you for that. So one of the things that we know about those brain circuits, those brain networks we're talking about, is that they are exceedingly flexible, meaning if the neurons themselves, the brain cells themselves, are very specialized. If one dies, it's not replaced, but the adaptation ability of those networks, the capacity to be modified by influence, to adapt, to react to changes, is very, very high. Yeah. Broadly speaking, we call that plasticity, the capacity of the brain to adapt and modify in response to different demands or different inputs. A sort of invention of nature to make the brain respond in a timely manner to the very rapidly world that we live in. And those mechanisms of plasticity depend on interactions between neurons. And by using TMS, transcranial magnetic stimulation, and recording the response in the human brain or in animal models, we can assess first of all, whether changes have taken place in circuits, plastic changes in response to an injury or otherwise. But number two, we can assess how well those mechanisms of plasticity work, how efficient they are. And a lot of my own work focus on those two questions. Can we demonstrate changes in the brain in response to learning? Can we potentially accelerate those mechanisms of learning by modifying the efficacy of the mechanisms of plasticity? And part of what we learned is not surprising, because there is a lot of evidence from other fields that the brain is quite plastic and that those circuits do change significantly, and we can demonstrate those changes. But more exciting, I think, we found out that we can assess the efficacy of those mechanisms of plasticity, how well those plasticity mechanisms work in a given brain, in a given person, in a given circuit. And in doing so, we learned, for example, that the efficiency of the mechanisms of plasticity changes in any one of us as we get older, that the response to the perturbation of TMS is quite specific for a given individual. There is a sort of fingerprint of a given subject, and that deviations of that individual characteristic may be a sign of disease, and that in certain illnesses, in certain conditions, part of the core abnormality is that those mechanisms of plasticity lack efficacy, either in specific circuits or more broadly. And so by characterizing plasticity and its efficiency, we get a core aspect of what accounts for brain health and what accounts for our changes of the brain as we age, identified and converted into a measure that we can ultimately even target with therapeutic interventions.
Now, one of the early applications of non-invasive electrical stimulation to a major neurological disorder was in depression, right, with electroconvulsive shock, which is it's very dramatic. It has beneficial effects. It induces a dramatic increase in neurotrophic factor levels in the brain, BDNF in particular. But it can also, it's improved a lot over the years. It can also lead to some problems with memory. What is the track record so far with repetitive TMS in people with depression?
Yeah, so you're absolutely right. Electroconvulsive therapy was one of the first sort of established electric or electromagnetic sort of therapies, and it's a very effective antidepressant, but it lacks selectivity. It's not just one brain circuit that is affected, and perhaps because of that, it leads to other potential problems. TMS allows to do a modulation of a brain circuit in a much more precise way. The way I like to think about it is that if I want to fill my mug of water, just selectively, I could put a dropper in here and fill it just to the right level, and it would be exquisitely precise and selective. And instead, what we often do currently in medicine is open up the ceiling of this room, let it pour in here, it fills in my mug, it makes me completely wet, it ruins a lot of things in the room. We call all that side effects and move on. And so the promise of TMS is to have that precision, that selectivity. When applied in depression, it required understanding what is the circuit of the brain that is altered in depression. And thanks to pioneering work from a large number of people, and the translation of that pioneering work into neurophysiologic interventions, including from people like Mark George and others, allowed to start showing that one can indeed modify those circuits in a very similar way as it has been shown that it can be modified with invasive stimulation, but in this case, non-invasively. And and that ultimately led to the approval by the Food and Drug Administration of a first device, and then since then a number of other devices have been approved.
And for depression, are they targeting the prefrontal cortex? We hear a lot about the prefrontal cortex, for example, psychedelics, right, are on the news now, have antidepressant effects, and one brain region they seem to impact particularly is the prefrontal cortex. Is that what's been used mostly for depression?
Yeah, that is exactly right. So the way to think about it, or the way I think about it, like I said, is that for any modulation of a brain circuit, we need an entry point, right? Traditionally, we need a window of entry. And in the case of depression, that circuit is broader than just the prefrontal cortex. But for non-invasive stimulation, it appears that the dorsolateral prefrontal cortex, maybe the ventrolateral, is the window of entry to the appropriate circuit. And the reason why I find it useful to think that way is because it also implies that if we have a way to target multiple ways, multiple points of entry to the same circuit, the effect of control of the circuit will be greater. And a lot of the field is developing now multifocal approaches to not just modulate one spot, but multiple areas in just the right way to maximally modulate a circuit.
So you mentioned they have a coil, right? And you're passing current through there like very quickly. But you know, I've looked on the web, and people can buy their own home TMS. Now, some of them are from China. I don't know about the quality, but the way they look, there's this little hat, and does it have multiple coils on it? Is that what is going on there?
So that's a great question too, Mark. I think one thing worth clarifying is that TMS, transcranial magnetic stimulation, is only one of several ways of non-invasively modulating the brain. And a lot of what people see and see on the web and do on their own, do-it-yourself kind of movement, uses a different technique called transcranial current stimulation. So it's different. But that said, there are magnetic stimulation devices also available, and some people have done it themselves and experimented on themselves. I get very worried about those at times, because there's lots of questions that we simply don't have the answer to. We were talking earlier, what exactly are the circuits that we're modifying for a given goal? What exactly are the substrates of that effect, the neurons that are being modified, and how much and for how long do you modify it? Lots of questions we don't know. I think it's important to be cautious about these techniques, because what we cannot pretend is that they have no effect. If we can modify the brain for the good, it is possible that sometimes it may go the wrong way.
Yeah, and maybe, you know, towards the end, we'll talk a little bit about the current healthcare system and problems with it, and this idea of lifestyle medicine in the digital age, and how the healthcare system can work with maybe not just the doctor, primary care doctor, but have some intervening person that helps the patient more directly, or, you know, there's apps and so on. But well, maybe I should just go to this. You had you know, I've had a longstanding interest in brain aging and Alzheimer's disease, and you did a recent pilot study in patients with early Alzheimer's, was it?
Yeah, so one of the things that we learned, let me maybe, if I may, give you a little bit of background as to how I got into this. So first of all, I am a behavioral neurologist. I'm a cognitive neurologist. I take care of patients with dementia. So that's been my interest, but clinically. But in addition to that, in the research studies I was talking about, exploring the efficacy of the mechanisms of plasticity, one of the things we found out is that as we all get older, the efficiency of the mechanisms of plasticity goes down. Yeah. Our brain is still plastic, but it's not as readily modified as it is in a younger age. And strikingly, we found out that in certain conditions, particularly in people with diabetes and in people with very, very early Alzheimer's disease, the brain was particularly non-plastic, particularly slow in being modified, and selectively particularly slow for what we call potentiation. So not for all types of modulation of the synapses, but of the connections between brain cells, but specifically for those that upregulate the efficiency. So we found that rather interesting and asked them the question, what if we use brain stimulation not just to measure, but to try to restore some of that capacity? Could that lead to a benefit? And of course, the question then becomes, well, a benefit for what? So you would want to do the stimulation, modify that property, and immediately after engage the brain in the kind of cognitive task, the learning task that is controlled by that circuit. And so that led to a number of studies asking the question, can we use TMS or later on ACS, a different form of non-invasive stimulation, but in both instances, in all instances, with the goal of modifying a brain circuit, specific brain circuit, making it more readily plastic, more readily engaged and modified by learning, and coupling the brain stimulation with an appropriate cognitive task, and see if we can restore some of the function that is lost in diseases like early dementia.
So there's really strong evidence that people who exercise regularly and don't overeat and also keep their minds intellectually challenged, socially challenged, are less likely to develop dementia and have a longer brain health. And that's consistent with that you're finding in diabetes, people with diabetes, right? It adversely affects the brain and accelerates age-related cognitive decline.
Yeah, there's a lot of epidemiologic data that a number of things, from diabetes and other illnesses to sedentary behavior and poor sleep and poor nutrition, a number of lifestyle factors and a number of chronic illnesses can increase the risk of age-related cognitive decline. Yeah. Now, one caveat is that we don't know that they do so on the basis of the same mechanisms. It may be through completely different mechanisms. But we do know in the meantime, thanks again to wonderful pioneering work for many scientists, among others, Mia Kivipelto and others, that interventions that modify some of those factors, engage people in modification, in more exercise, in better nutrition support, that those interventions actually translate into a reduction in risk of these illnesses. That's super exciting. And there is a whole body of work aimed at modifying lifestyles to reduce and prevent or delay at least the risk of dementia. We've been interested in the question of, can one understand the mechanisms with some of these brain stimulation techniques, and can one accelerate the benefit by combining, not substituting, but combining lifestyle modifications with this type of neurophysiologic interventions that modify specific circuits?
There's a lot of evidence that across a lot of different brain disorders that there's problems in neural circuits constraining excitability such that there's overexcitation. That's obvious in epilepsy, but it's also true in stroke where if the neurons aren't getting enough glucose and oxygen and they're continuing to be stimulated excessively, they're more likely to die. And we think it's true more insidiously in Alzheimer's disease too, that there's this cytotoxic component. One thing we found, towards the end, but right before I retired from the NIH, we did a study. We found that intermittent fasting, which extends lifespan in rats and mice and seems to do good things all around, it enhanced GABAergic tone. That is, it enhanced the ability of the circuits to constrain their activity within normal, presumably healthy limits. Now, I think these GABAergic interneurons are very interesting because they fire like crazy. They're very active. They have a high metabolic load, high oxidative stress. And so just intuitively, that would make sense that particular characteristics would make those neurons vulnerable to aging, which involves problems in controlling oxidative stress, energy metabolism, and so on. Is there any way to get, I come back to the same question, is there any way to get at pharmacologically or otherwise get at this issue of these GABAergic neurons?
Yeah, so using brain stimulation by doing measures of the response of the brain to a single or a pair of pulses, so something we call pair pulse stimulation, one can explore selectively or relatively selectively inhibitory versus excitatory connections and look at intracortical inhibition versus intracortical facilitation circuits and show deficits in one versus the other. And of course, one can, on the basis of those measures, calculate an excitation inhibition balance. To be clear, we don't have the selectivity of specific GABAergic neurons, but one can get a relatively precise view of that excitation inhibition balance, at least longitudinally over time in a given person. Sorry. No,
yeah, in previous episodes of this podcast, I've had Rob Malenka and others, talking about LTP and LTD, and it's worked out in animals. If you stimulate pathways at a certain frequency, it will strengthen the synapses. If you stimulate at a lower frequency, it can inhibit. And my understanding is with TMS, you can do that. You can vary the stimulation and kind of mimic those patterns that seem to be involved in learning and memory, or maybe impairment of learning and memory.
Yeah, it's a good point. So you can modify the frequency of the stimulation with TMS, with repetitive TMS. We can apply slow or faster stimulation. We can apply stimulation in continuous trains or intermittent trains, much like we can do in a slice preparation with electric stimulation. And the big difference with what Bob and many other basic scientists do compared to what we can do in humans is that in their case, they know exactly what stimulation they're applying to exactly what connections, and they're measuring from those connections and those connections alone. We don't have that selectivity, but remarkably, despite the lack of selectivity, it appears that the brain, in response to the intermittent or continuous, fast or slow forms of TMS, responds in a similar way to what it does in a slice preparation. So we often talk about LTP-like and LTD-like responses. We know that it's not the exact same physiologic mechanism in the brain, but it has commonalities with what one finds in a slice preparation, including the fact that there is a buildup of the effect. There is a dependency on the pattern of stimulation and the frequency. And one can combine both of those principles to induce therapeutically, potentially, a potentiation or a suppression of the effect. One can apply the depressing, quote unquote, patterns of stimulation to control a seizure focus, and folks are exploring that, and one can apply the potentiation parameters and accelerate learning and potentially accelerate recovery from stroke and other illnesses.
Is there any, most effort, as it should be, is on neurological disorders and enhancing recovery or slowing decline, for example, in the case of Alzheimer's? Is there any interest? I'm sure there are there's a lot of very health conscious people that do this biohacking stuff, right? And try to enhance their cognition and make themselves more productive. Is that, and I'm sure there's companies that are picking up on that and trying to get people to spend their money, but is there any evidence that in a say a young healthy individual that TMS can enhance any brain function?
It's a great question. So there is indeed a lot of interest in brain enhancement. Again, the kind of stuff that makes me very leery sometimes, because there is a very slippery slope on both on ethics and on who decides what is appropriate to enhance or what not. But in very careful experimental settings, it has been demonstrated that you can speed up acquisition of skills, you can improve consolidation of knowledge, you can make people learn a 5-finger movement exercise quicker. Masahiro Kobayashi showed that in my lab working with me a number of years ago, and you can do so in different ways depending on exactly what components of a given network you modify in what direction.
And so the TMS is applied while they're learning?
In this particular set of experiments, in general, just before. So you modulate the circuit and then you engage the circuit in a given learning. But to your point, you could apply other forms of stimulation in similar ways. But the problem with TMS applied during the learning is that there is a disruptive effect on the brain by the tapping sensation and so forth. So in general, it's done before. But the big issue in all this field is that we don't know yet that the brain is not a net zero-sum operator. So it is possible that you improve one thing, and that one thing has a cost, and you're worsening something else. And in that risk-benefit ratio balance in somebody with a given illness that is disabling is one thing. To do the same intervention in somebody who is arguably functioning fine in order to improve one aspect, potentially at a cost of something that we don't quite know what it is, the cost would be, that's a very different, sort of ethically very different decision process. And so I think, again, it's something that needs to be done with appropriate caution and links to the question you were asking before of what about people just doing it, including companies. And I think there is a need for a neuroethical approach to technology, to neurotechnology, because the advances in neurotechnology are fast, are vast, are exciting, but need to be, we need to limit our own sort of desire to advance the field.
And how does FDA, do they even regulate any of these devices at all?
Yeah, the devices are regulated as medical devices. So where it becomes a little slippery is, I mean, where it becomes a potential loophole is when things are developed as direct-to-consumer devices, and
Right.
there is no medical intention, but a more sort of cosmetic intention. And I think that that's part of the neural rights kind of movement that is necessary, I think, to become aware of the advances in neurotechnology, exciting as they are, need to call for appropriate addressing of potential misuses that could happen.
And that, to finish up, you had a recent paper on intelligent technology therapy assistant. What is that?
Well, I think that one of the exciting things about neurotechnology and about technologies in general is the ability to read brain activity in a variety of ways, from EEG or from behavior metrics, and using artificial intelligence extract very precise metrics, very detailed insights into the pattern of behavior, the characteristics of individuals, essentially converting to quantitative metrics the impression that an expert will have, right? There is this notion in neuropsychology called the Boston process approach. If I observe you doing something and I'm really good at it, I can get a sense of how well you're functioning by leveraging the focusing on the process rather than just on the output of the end result. So imagine using technology to do that. If we use technology to do that, we can generate very precise and sensitive metrics that provide quantification to impressions of people, such that now the tools are available for the experts to gain insights faster, quicker, and more precise. And so the idea behind this sort of technology-enabled assistant is to not just have somebody observe, but use technology to capture insights that we can then use. So to close the loop with brain stimulation, imagine that we are capturing exactly how I'm performing, what I'm saying, how I'm saying it, the hesitations, the pauses, the brain activity associated with it. And when I use brain stimulation, I use that vast body of information quantified to define when to stimulate, for how long to stimulate, and when to stop. It becomes a closed loop feedback system that optimizes the stimulation for a desired effect.
So you get rapid, unbiased feedback and adjustment accordingly.
Exactly. So a very personalized, very precision medicine approach, but in a different notion of precision medicine, not from genetics, but rather from the pattern of activity, the state of activity, and optimizing that for a given desired outcome that needs to be empowered and defined by the person. There's the need for the neural rights that we were talking about.
Yeah, for viewers and listeners, if you want to know more about Professor Pascual-Leone's work, you can go on PubMed and put in his name and plus the word review is what I would suggest, and then you can get reviews. Most of these are publicly available now, which is a good thing. I will put links to some of them in the description section of this podcast. And thanks for taking the time. You're incredibly busy, and I appreciate your time. And I hope the listeners and viewers learn about this. And it's a huge, very important area, very exciting. These methods are, for the most part, perfectly safe, at least when used, in the clinic, they're perfectly safe and non-invasive. And, so side effect problems with drugs is a big issue, right? And
yeah, drugs is a big issue of side effects. I think to your point, these tools can be perfectly safe. Can be is the operating word here. If used appropriately with folks who know what they're doing and have the appropriate competencies, and where the goal and the target is well defined, I think that's very powerful and very promising technology. But by the same token, it's like a knife, right? You can do great things with a knife. You can really do bad things, and so I think it's important to
yeah, but as the technology, so you can imagine. So right now, say someone has a mental disorder and they're taking some drugs. They're supposed to keep in communication with their psychiatrist, but there's a lot of gaps in there. And if you had something where you had a treatment that there's a system for feedback and correction, and then maybe the doctor is monitoring, or someone in his office monitoring this information that's being generated in real time from the patient.
Absolutely. Yeah. So I'm obviously a big believer in these techniques. Obviously devoted a lot of my career to developing them and to apply them. And I think there is lots of opportunity, precisely like you're saying, to define disability, to empower the modulation of a disability, of the brain activity that accounts for the disability, and overcome it. And that becomes relevant for developmental disorders all the way to geriatric populations, and I think that's super exciting to be able to alleviate people's sufferings and to promote functional states in all of us.
Yeah. Well, thanks again. Take care.
Thank you so much, Mark. I appreciate your interest. Thank you.
Yeah. Bye.